Sathyabama Institute of Science and Technology.
A very good day, everyone. Myself, Preetha Wilma Dawson, Assistant Professor, College of Physiotherapy, Sathyabama Institute of Science and Technology, deemed to be a university. And today's topic is about visual processing. All the senses in our body gives us vital information about the surroundings and the environment. But the one we mostly rely on is vision. The sense organ for vision is a biological instrument which helps to turn the light into brain's language of electrical signal. The sense of vision is a process of converting the unstructured data into a meaningful image. So, the light energy from the external environment comes in the form of electromagnetic radiation, and then it is getting converted into electrical signals. These electrical signals are carried to the higher centers for further interpretation and recognition. Next, we'll discuss about visual processing in a flowchart. The light from the external environment will be reaching the eye. From there, it will go to the internal layer of the human eyeball. It is a light sensitive structure which is called retina. And the retina contains photoreceptors. Namely, we have three photoreceptors. The first one, the rods, this is responsible for vision. And the next one is cons. This is also responsible of vision. And next one comes the photosensitive retinal ganglion cells. These receptor cells helps in the regulation of circadian rhythm. First, we'll discuss about rods. Rods are cylindrical structure. The length is about 40 to 60 mu and diameter is about 2 mu. Each rod is composed of four structures: the outer segment, inner segment, cell body and synaptic terminal. First, we'll start with outer segment. Outer segment of the rod cell is long and slender, and this gives them a rod-like appearance. The outer segment of the rod cell is formed by modified cilia and it contains a pile of freely floating flat membranous discs. There are about 1000 discs in each rod, and the disc cells of each rod contains a photosensitive pigment called rhodopsin. Next, we'll discuss about inner segment. There is a structure which connects the outer segment and the inner segment. This structure is called as modified cilium. The inner segment contains many types of organelles with n number of mitochondria in it. Next comes the cell body. The enlarged portion of the rod fiber is called cell body and it possesses a nucleus. Next comes the synaptic terminal. Synaptic terminal is a thick fiber which is arising from the cell body and then passing through the outer plexiform layer. So this outer plexiform layer is the interconnections of neurons, and from there it ends in a small and enlarged synaptic terminal or synaptic body. So we have synaptic vessels present in the synaptic terminals and it contains a neurotransmitter called glutamate. This glutamate is a excitatory neurotransmitter. Next, we'll discuss about the functions of the rods. The rods are very sensitive to light and they have low threshold. The rods are responsible for dim light vision or night vision. That is, imagine when you are in a dark room or during night time, you are able to feel some visual perception. This visual perception is completely taken over by the control of rods. So this type of vision is called as scotopic vision. The rods mainly, they will not take part in resolving detailing about visual acuity. Visual acuity is the boundary of images and also they do not take part in vision color. The vision by rod is black, white or grey in color. So we have a photosensitive pigment in rods that is called as rhodopsin. This rhodopsin is also known as visual purple, which is responsible for scotopic vision. We have a picture representation of rods. Let's see about it. First comes the outer segment and the inner segment, and we have a structure connecting the outer and the inner segment is called as connecting cilium. And we have a synaptic terminal which is small and the cell body which possesses a nucleus in it, and we have a disc inside the outer segment. Next, we'll start with cones. Cones are the visual receptors. The length is about 35 to 40 mu and the shape is flask shape. The cones are formed by four parts similar to rods: the outer segment, the inner segment, cell body, and the synaptic terminal. First, we'll start with the outer segment. Here, the outer segment is a small conical structure. In rods, we discussed that the outer segment consists of a pile of free-floating flat membranous discs. But in the cones, we do not have any disc-like structure, but we have infoldings of cell membranes. This infoldings of cell membranes is called as sacules. Next comes the inner segment. The outer segment and the inner segment is connected by a structure called modified cilium. It is very similar to rods. Next comes the cell body. The cone fibers arising from the inner segment is very thick in cones and they possess a nuclei in it. Next, we'll discuss about synaptic terminal. Fibers from the cell body leaves from there and enters into a plexiform layer. We have discussed about it earlier. Plexiform layer is the interconnections of neuron. From there, it will be having an enlarged structure which is called as synaptic terminal. Next, we'll see about the functions of cones. The cones are only sensitive to very bright light and they have a high threshold. The cones are responsible for bright light vision and also day vision. So let's consider when we have an area with well lightened structure, so the vision we are able to perceive with it is regulated by the cones, and the vision we are able to perceive in the daytime, this action is regulated by cones. Cones are responsible for acuity, that is the boundary of the image, and also for color vision. The photosensitive pigments in cones. So in general, we have three photosensitive pigments in cones, namely porphyrosin, iodopsin, and synopsin. These are the three photosensitive pigments which are present in cones, and we have a protein called photopsin which is present in the cones. Next, we'll see about the structure of cones. We have the outer segment, and the outer segment here does not contain a disc. We have a saccule, that is the infoldings of the cell membrane. And then we have the inner segment. The structure connecting the outer and the inner segment is called as connecting cilium. And then comes the cell body, which possesses a nucleus in it, and then comes the synaptic terminal, which is enlarged here. Now we'll discuss about the third photoreceptor, that is the photosensitive retinal ganglion cells. These photosensitive retinal ganglion cells are mainly responsible to regulate the circadian rhythm and also to maintain the pupillary reflex. So these photoreceptors will be responding to light in the absence of rods and cones. Many studies have proved that a small percentage of mammalian retinal ganglion cells expresses the photopigment called melanopsin. The melanopsin is nothing but a G protein coupled receptor. As I have discussed before, this photosensitive retinal ganglion cells is responsible to maintain and regulate circadian rhythm. Along with that, it is also responsible for pupillary reflex. So next, we'll discuss about the circadian rhythm. Circadian rhythm is a biological process that displays an endogenous entrainable oscillation of 24 hours. So, even though the circadian rhythm is an endogenous one, that is a self-sustained one, we are able to adjust this endogenous scale in it. We are able to adjust this endogenous component according to the external environment, that is the external cues the environment giving us. So the external cues are called as zeitgebers. The most important zeitgeber is the daylight. So let's see the flow chart about it. Circadian rhythm, it's an internal clock which is present in the brain. This works in response to light. So it gives two responses and maintain these two responses. One, it also maintains the sleep cycle, along with that to maintain the alertness in an individual. Next, in general, we have studied about what is rods, what are the structures, what are the components and functions of the rods. On the other hand, we also studied about the structure, function and the components of cones. Now, let us compare and differentiate the structure, function and the components of rods and cones. So, first we'll discuss about the length. The length of rod is about 40 to 60 mu, and for cones it is about 35 to 40 mu. The diameter is about 2 mu and there it is about 5 mu. The shape is cylindrical in rods and the shape is flask in cones. The outer segment in rods is long and slender, whereas the outer segment in cones is small and conical. The sensitivity of the rods, they are more sensitive to light, but whereas cones are only sensitive to a very bright light. The threshold is low in case of rods and the threshold is high in case of cones. The type of vision perceived by rods is called as scotopic vision and the type of vision perceived by cones is called as photopic vision. Acuity and vision. Rods is not responsible to maintain visual acuity and vision color, whereas cones are responsible to maintain visual acuity and visual color. The pigment present in rods is called as rhodopsin. The pigment present in cones is called as phoropsin, iodopsin and cyanopsin. Number of rods in each eye is about 12 million and number of cones in each eye is about 6 million. Now we'll have a quick recap. First, we discussed about what is sense of vision. Sense of vision is nothing but the process of converting an unstructured data into a meaningful image. Next, the process of visual processing we have discussed in a flowchart, that is, from the eye, the visual input is getting into the retina, which is the light sensitive structure. From there, we have a photoreceptors present in the retina. So, the photoreceptors in the retina, one is rods, cones and retinal ganglion cells. After that, we have detailedly studied about the structure, functions and the components of rods and cones as well. Then we had an overview about what is photosensitive retinal ganglion cells and the major regulation of it is about circadian rhythm, and just we had an overview on it. So this was the topic covered for today. Thank you.